lipopolysaccharide is MD2. Now, what happens if bacteria, now if you go back to the microbiota, if you remember the mucosal immunity, we said there is a big group of gram-negative bacteria, the largest group, the Bacteroides, they have a penta-acylated LPS. And they have a lipopolysaccharide which does not have 6 fatty acid, it has 5 fatty acid chain. Is this a potent activator of TLR4?
No.
No, it doesn't activate.
It doesn't activate it well. And if you go to the cartoon, this 6 chain here is absent, which cross-link the 2 receptor. It means that this lipopolysaccharide is a very poor activator of TLR4. And some bacterial pathogens like Yersinia pestis, for example, which causes plague, and Helicobacter, and that causes the infections of the stomach, they have a tetra-acylated LPS. And they even have only 4 fatty acid, which means that binding to MD2 is already poor. Okay, so they escape detection by TLR4. So, and this is the importance of these structural studies, is that they told us that bacteria which try to reduce the number of fatty acid chains will produce a poor dimerization of TLR4 and poor signaling from that receptor. Okay. Now, how do toll-like receptors signal into the cell? Now, toll-like receptors basically, or signals generated from toll-like receptor, activate the following responses: an inflammatory cytokine production, chemokine production, antimicrobial peptides, and type 1 interferon. Okay, so inflammatory cytokines, chemotactic factors, AMPs, and the type 1 interferons, interferon alpha and beta. Out of these 4, which of these do you think is the most evolutionary conserved target of TLR signaling? If I give you this question, out of these cytokines, chemotactic factors, AMPs, type 1 interferons, which one is the most evolutionary target of TLR signaling?
AMP.
Why AMPs? It is AMPs, but why?
Because موجودة بالinvertebrates والvertebrates.
Exactly. If we take insects where tolls were discovered, like Drosophila, toll-like receptor signaling, which occurs in can be in the epithelia, but also in specific cells, we call them fat body cells, which are similar to our liver, controls AMP genes. So the main immunity genes controlled by TLR signaling are the antimicrobial peptide gene. Okay? And this function of TLR controlling antimicrobial peptide gene remain conserved in mammals and vertebrates in general. Okay? So TLRs are not only found on our immune cells, they are also found on epithelial cells. Okay? So for example, you have TLR4 is expressed on epithelial cells, but TLR4 is also expressed on macrophages. Do you think that if I activate TLR4 on epithelia and TLR4 on macrophages, I get the same set of genes activated? So TLR4 is found on different types of cells. And depending where it is found, it can activate a different set of genes. Okay? And there is different specificities for different cells. So TLR on epithelial cell maintained, if you want, its evolutionary conserved role in regulating antimicrobial peptide genes. Okay? But also our epithelial cells, I know there is inflammation, they can produce some cytokines. But TLR on phagocytic cells or immune cells, they can trigger many other responses. They can trigger mainly cytokine production, but also they can enhance survival. They can control the survival of the cell, reduce the expression of pro-apoptotic genes, and so on, as we have seen for neutrophils. Okay? So the same toll-like receptor on different cells can have different outcomes. Okay? And this is important to keep in mind. Now, how do the TLRs signal? TLRs use 4 adapter molecules that bind to their cytoplasmic domain. You can see them here in the figure: TRAP, MID88, TRAM, and TRIF. Okay, these 4 molecules. But TRAP and TRAM, they are, if you want, adapters to the adapters. Okay, so these are, for example, TRAP is needed to recruit the second adapter, which is MID88. What is an adapter protein? It's a protein which has no enzymatic activity. It is just there to recruit another protein called an adapter protein. So TRAP is there to recruit MID88, and TRAM is there to recruit TRIF. So if you look at the downstream adapters, it is either MID88 in most of them, or TRIF in two receptors, TLR4 and TLR3. So what starts the signaling pathways is either TRIF or MID88. So we have two signaling pathways downstream of TLRs, a MID88 dependent pathway or a TRIF mediated pathway, because these are the two adapters which start recruiting signaling molecules. And what is the outcome of activating the toll-like receptor signaling in terms of the type of transcription factors produced? It is three main transcription factors, NF-kappa B and AP1. And these two transcription factors are the ones that control inflammatory cytokine production and the production of chemokines and AMPs. And the IRF, or interferon responsive factors 3, 7, and the IRFs will control the expression of type 1 interferons. Okay? So these three transcription factors basically are the key transcription factors activated downstream of the toll-like receptor. Now, all TLRs have a signaling pathway that is either TRIF or MID88, except TLR4, which can use both. It has both signaling pathway, MID88 and TRIF, but it cannot use them at the same time. MID88 is used when TLR4 is on the surface, and TRIF is used when TLR4 is in the endosomal compartment, as we'll see in a second. Any question on this figure?
No.
Okay. So I will give you one example of signaling only, okay, because signaling is very complex and you spend the whole semester on it if you want. But let's see, how do these TLR signal and what do we learn from signaling processes in this? So if we look at activation of NF-kappa B transcription factor by toll-like receptor 4. Now, TLRs dimerize, we said, and this brings their signaling domains close to each other. Okay? When these signaling domains are close to each other, the MID88, which is recruited by each receptor, you see it in red, eventually form a complex, a cluster around the receptor. Who do you know, who remembers what is the name of the cytoplasmic signaling domain of TLRs? How do we call it? It's a famous domain in the cytoplasm. It is similar
RTK.
Sorry?
RTK.
No, it is similar
No.
to the interleukin-1 receptor signaling domain.
Decoupled protein.
No. It's called the TIR domain. TIR domain. TIR. Why? For toll interleukin-1 receptor-like domain, because it's a domain that is very similar between toll and interleukin-1 receptor. So the interleukin-1 receptor recruits very similar complexes downstream, similar to the TLR. So this TIR domain that is found here is also found in MID88. So MID88 can bind to this TIR through TIR-TIR interactions. And once MID88, which is an adapter protein, is recruited, it recruits the kinase. We call them here IRAK4 and IRAK1 for interleukin-1 receptor-associated kinase. Okay, so IRAK is interleukin-1 receptor-associated kinase because the first time it was discovered, it was underneath of the IL-1 receptor, and later they found that it acts downstream of several receptors. Now, when this complex forms, IRAK4 recruits, IRAK4 and IRAK1 are recruited to the receptor. IRAK4 is a kinase. It autophosphorylate and activate itself. So eventually, IRAK4 will phosphorylate IRAK1. And this IRAK1 here recruits a complex of ligases, ubiquitin ligases, TRAF6, UVE. You don't have to remember the names. You just remember ubiquitin ligases. So IRAK1 recruits TRAF6, okay? And eventually TRAF6 through many other E1, E2, and its own E3 ligase activity, ubiquitin ligase activity, it ubiquitinates itself. Okay? And in addition to its, you see a ubiquitin chain here, in addition to ubiquitinating itself, it recruits or it interacts with a kinase known as TAC1. And once TAC1 comes close to TRAF6, IRAK1 phosphorylates TAC1. So now TAC1 becomes active. Now, I will go to the end and then I will tell you what's the significance of these stories all. So now that TAC1 is a kinase, is active, in fact, so TAC1 is activated by a phosphorylating event, but also by binding to the ubiquitin chain on TRAF6. This complex now, TAC1, TRAF6, leave the receptor, goes to the cytosol to activate the NF-kappa B transcription factor. Now, what is NF-kappa B? I don't know if we discussed this before. NF-kappa B is found in the cytoplasm. It is a dimer made up of P50, P65, two proteins, and it is locked by an inhibitor known as I-kappa B. Okay? So I-kappa B is an inhibitor of NF-kappa B that inhibit the protein from going to the nucleus. Okay? So in order to free NF-kappa B, we need to phosphorylate I-kappa B. I-kappa B dissociate, NF-kappa B can move to the nucleus. Okay? So in order to phosphorylate I-kappa B, I need a kinase to be activated, and this is the IKK, or the I-kappa B kinase complex that you see here. It's made up of these two kinases, alpha and beta, and the gamma, which is we call it NEMO protein, is holding these two kinases. Okay? So IKK alpha and beta, specifically the beta subunit, will phosphorylate I-kappa B to release NF-kappa B. But to activate this kinase complex here, I need another kinase, which is TAC1. Okay, so I need to bring TAC1 very close to this IKK complex so that TAC1 phosphorylates the orange circle. There is a mistake, so it should be on the orange circle. And the orange circle phosphorylates I-kappa B so that NF-kappa B moves to the nucleus and activate the inflammatory genes or AMP genes. Okay, so again, you see that the interaction here.
Doctor?
Yes.
We need to join other classes.
You need to?
Join other classes.
Okay, so I'm done, I think. So it's just, there is 1 minute more.
Oh, okay.
Okay, so why do we need these ubiquitin chains here? And I'll come next lecture, I'll discuss what type of ubiquitin, because these are not ubiquitin chains that lead to protein degradation. They are ubiquitin chains involved in signaling. So I need the ubiquitin chain here to bring this kinase in close proximity to this IKK so that they can eventually phosphorylate each other. Okay? And without such chains or platforms, these molecules can never find each other in the cytosol. Okay, so the whole signaling pathway is built around how the transcription factor needs to be activated. And in this case, activation of NF-kappa B needs a phosphorylation of its inhibitor, which is found in red here. And this is why I need the kinase IKK, and I need another kinase to phosphorylate it, to activate it. And to activate TAC1, I need the IRAK1 kinase to phosphorylate it. Okay, so the first complex here leads to the TAC1 phosphorylation. The second complex here leads to IKK phosphorylation and eventually NF-kappa B activation. Okay, so if you have no questions, I will stop here for today. And next time I will discuss what type of ubiquitin signaling is taking place in these cells. Any questions?
Sir, tomorrow the quiz is at 6:30. You'll send it by email?
So I will send it to you by email.
Okay, thank you.
Okay.
Thank you.
Okay. Have a nice day then.